Alright, so we're going to talk about amino acids here. So we'll introduce them, and then we will talk about the structure and a few other details here and there. But this will be kind of an introduction of just the monomer. So in the next video, I'll go into more detail on some key features for these monomers, and then after that, we will talk about how they polymerize and form a chain. And then in the next section, we'll talk about how that chain becomes a protein and the key features of proteins. So, amino acids are, if a protein is made from a chain, amino acids are the link in the chain. So it's made up of amino acids. So these are pieces that link up to form what we'll call a peptide chain, and then that peptide chain can become a protein. So the difference between that long noodle, that chain, and a fully formed protein is a big difference. And so we'll have a whole section where we talk about that. And if you've taken nutrition, you might already know what an essential amino acid is. But essential amino acids are ones that we cannot metabolize ourselves. So we need amino acids because we use proteins a lot for all of our cellular functions. It can be anything from regulating blood sugar to carrying oxygen around the body. So hemoglobin is a protein, and it's involved with carrying oxygen through the bloodstream. All of these use amino acids. All these are proteins, they're made of amino acids. Now we have the ability, if we're deficient in amino acid, if we need more, we can produce it, with a few exceptions. We're not going to worry about which ones are which, but if we can't produce it ourselves, then we have to get it from our diet. And so those are what essential amino acids are. And I think that's covered more in like a nutrition class or something like that. I'm not going to worry about it too much. But basically anytime you have anything like essential vitamins, minerals, amino acids, it means we have to procure them by eating. We have to take them in from our environment. We can't produce them ourselves. So it means it's needed in the diet, because we just can't make it. Our bodies are pretty good about, if we can do something, we do. And if we can't, well then, we figure out what can and we kill and take from that. That's what we do. That plant over there, it's got some stuff I need, mine. I can't photosynthesize. I can't turn CO2 into sugars and starches and stuff, but that plant can. But I need those sugars and starches to survive. Goodbye, plant. Hello, calories. That's what we do. And I think probably the biggest key to world peace will be when we have little green people walking around, and they just photosynthesize, so they don't need to eat or anything. Like, you don't really need anything. Like, you're hungry, you just go lay in the sun. And so what you'll have is, like, in the southern part of the country where there's more sun, we'll have a big obesity epidemic. But up here in the Pacific Northwest, rock solid. We'll be totally healthy, because for a few months out of the year, we're starving. That's what we do. It's like, oh, holiday season's coming along. Better not, overeat. Oh, wait, I can't because it's cloud cover. Anyway, that's just my own little goal in life, is photosynthesizing people. That's what we need. That would solve all of the world's problems. And then all you, like I said, you just lay out. You just find, a spot by a pool or something, go lay out and take a nap, wake up full. What more could you ask for? Alright, so let's talk about the structure next. So I'm just going to draw a generic structure of an amino acid. The name kind of tells you the pieces in it, at least the key pieces. So we're going to have an amine group, or also known as an amino group. We've got a carbon in the middle here. And then we have a carboxylic acid. And so off this carbon there's always one hydrogen, and then I'm going to put an R here. And R is just kind of a stand-in. It's a wild card. It could be an H, it could be a long carbon chain, it could be all sorts of stuff. So it just means that this carbon is bonded to more stuff that's of an organic nature. But we've got kind of three pieces here. So we've got an amino group, we've got a carboxylic acid. And they're not right next to each other because in the middle we have what's called the alpha carbon. So this is the Greek letter alpha right here. And off of the alpha carbon we have what's called a side chain, which is exactly like what we talked about before. So this is like our main group. This is what defines an amino acid. Amino acid. Amino acid. One carbon in the middle, and what makes amino acids different from each other, there's over 20 different varieties, is just what R is. Like I said, it could be a hydrogen, it could be a CH3, it could be a carbon with an alcohol group, it could be a chain of carbons with some other stuff on them, but this side chain differentiates between different amino acids, but they all have this same repeating structure down here. This is like the backbone, this is the bread and butter. If it doesn't have this structure, with whatever's coming off of it up here, but this piece right here, then it's not an amino acid. So, the next video we're going to go into more detail on the side chains. There's important properties. At no point will you have to memorize any specific amino acid structures, but you should know the general. How do you know an amino acid when you see one? Right here. This piece tells you. So just like you look for rings with nitrogens in them, maybe a sugar phosphate backbone, and you can tell that you have a nucleic acid. Well, with amino acids, you just look for these pieces right here. Doesn't matter what R is. All that matters is you see this. You see this? Amino acid. Amino acid. That's what you got. So, one more detail we're going to talk about with the monomer. This is always kind of a tricky one. So, individual amino acids is the effect of pH. We'll talk more about pH later, but it does have a very important effect on these amino acids. If it didn't, I wouldn't talk about it. You can trust me on that. We've got plenty of other stuff to talk about in this class, but this is a very important detail, especially for the next video when we talk about side chain properties. And so what we have is the fact that they are affected by pH. And what we get is something like this. So we can have H3N plus, so we get an extra H plus on our amino group. It can become a cation. So first I'm just going to sketch out the whole thing here as we go through the pH scale. It's supposed to be at low pH. And then I have these little arrows that means these are interchangeable. So as pH changes, it starts transforming into this form. And so we still have a plus on the N here with an extra H. This is something that it does. Have an R group off of we have our side chain off of our alpha carbon in the middle. But now our carboxyl group is an anion. It loses an H plus. As the pH starts to increase, H's start popping off. And so if we go further, what we get is the following. It's no longer a cation, it's H2N, CH, and then COO minus. So the carboxyl group looks just like it did in the middle. And this is high pH. Okay. Now, this middle one right here, this is the one that we see in cells. This is the form at physiological pH. So this is what they look like when they are floating around inside of us right now. So you have a part of, if this is just an individual amino acid, you have your amino group being a cation, and you have your carboxyl group being an anion. And there's actually a name for this. So when you have a molecule that has both a positive and negative charge, like full ion charges, not dipoles, but like full ion charges that are opposite but on the same molecule, this is called a Zwitter ion. So it has a plus charge and a minus charge. Again, this is different than a polar molecule because these are full charges. This isn't like a delta plus or delta minus or a little arrow. Like, this is a +1. Oh, sorry, this is a +1. Gotta watch my camera blocking there. This is a -1. That's what makes it a zwitterion. So it has a plus charge and a minus charge on the same molecule. And so this really helps it be soluble in the cell. We don't want these getting stuck to, say, cell walls. We don't want them to be oil soluble. We want them to be nice and water soluble. And this ion, as well as the fact that these are soluble functional groups on the ends, but this ionization helps even more so. And so these are floating around freely in the cell, and they're getting snatched up and they're getting put into proteins. Or your digestion system is breaking proteins up into amino acids, potentially. Sometimes chunks get reused. It can get a little tricky, but generally you break down whole proteins into amino acids, and you then form new proteins out of it. Or if it's your own protein and you're just not using it anymore, it gets damaged or it's just not necessary, then you can break down the protein. Sorry, I don't know if you can hear, but my dogs are barking because there's probably something trying to break into the house, like a 7-Eleven cup or whatever. That's not too far-fetched. Our second dog, we got her, and the first week we had her, she barked out the window at a 7-Eleven cup in the street for about an hour. I couldn't get her to stop. I don't think she has very good eyesight. But in her defense, it was moving with a light breeze, so that was very threatening. Especially since there was only a window to protect her from it, and like 20 feet of distance. Alright, so, thank God they're going outside now. They use a cat door, by the way, because they're chihuahuas. That's true. Like, people think we have cats. No, we don't have any cats. We just have a cat door that our dogs use. Okay, so, where was I before I was so rudely interrupted by those little monsters? So zwitterion, okay, so same molecule plus minus charge. Oh yeah, so it's very water soluble, and that allows it to move freely through the cell and get used for biological processes. So when we move forward, what we're really going to focus on in the next video is what this R group means. Because when we make this chain, so we'll talk about the R group, and then we will talk about making a chain out of them and then folding that into a protein. So maybe you've heard of the term protein folding before. Well, that's because proteins are these 3D structures. It's like you have a string and you got to tie knots out of it, and then you make like a 3D structure. That's what's happening with what we call the peptide chain. We make a chain of amino acids, and then it's kind of like it gets tied in knots, and you get a 3D structure. It makes like a blob, and that blob performs some type of function. Generally, we're not making proteins for no reason. It could be ion channels in the cell membrane. It could be some more DNA polymerase to copy DNA. It could be any number, insulin to help transport, glucose in and out of cells in the bloodstream. Any number of things you can imagine. Generally, it's a protein that's performing the function in terms of actually like mechanical doing things on a biological level. These are the little microscopic machines that handle everything. Alright, so I'll leave that for the future videos, but be aware. So the next one, we're really going to focus on R. So hopefully right now you're comfortable with, an amino acid when you see one, and just understand that these amine groups are going to be positive, these carboxyl groups are going to be a negative anion at physiological pH. And I'll give you a little hint into the future, that is the case for any amine and carboxyl groups, including if they're on the side chain, because we can have those groups on side chains. Okay, so that's it for now. So yeah, we'll focus on the side chain, and then we'll talk about the polymerization process.